Hi, this is Ahed with Logos Learning, and in this video we will be talking about fermentation or anaerobic respiration. Now let's talk about cellular respiration with sufficient oxygen to draw the contrast between that and what happens in low oxygen conditions. Now cellular respiration is a redox reaction. Redox, if we look at the word redox, it's composed of two little ones, red and ox, where red stands for reduction and ox stands for oxidation. So we call it redox because reduction and oxidation reactions are always coupled. A reduction reaction is a reaction where something or a compound, an element, gains electrons. So to gain an electron is a reduction. And these electrons had to come from somewhere. It's from the other species that's lost electrons. So if you gain electrons, let me write down here. If you gain electrons, it's a reduction. And if something loses electrons, it is an oxidation. Something who loses electrons gets oxidized. Now, cellular respiration is a redox reaction in which the glucose is oxidized to carbon dioxide. So here I said LEO, because the glucose oxidized, lose electron oxidation. And the oxygen at the end of the reaction is reduced to water. But this is at the end of the reaction. The electrons go elsewhere during the process. So you notice here that if glucose were to be oxidized, something or someone had to take the electrons. The electrons don't just go float in the outer space. Something had to take them, and that is the oxygen. So the oxygen is the electron receptor. But here we're going to say that it is a final electron receptor. It is needed for this reaction. Just like any other type of combustion reaction, oxygen is one of the necessary reactants. Now let's take a look at how I broke down cellular respiration for the purpose of explaining this process, reduction and oxidation. So the reaction, cellular respiration, takes place in a series of steps. The first one being glycolysis, which takes place in the cytoplasm. Glycolysis produces 2 ATPs and 2 NADHs. This is the reduced form of the electron carrier NAD+. Okay, so NAD+, once it gains electrons, it becomes NADH. So this one has electrons. Okay, NADH has electrons. Now, if you take a look at this here, in this reaction, glucose is converted to pyruvate at the same time that NAD+ is being converted to NADH. NAD+ to NADH is a reduction reaction because the NAD+ is gaining 2 electrons to become NADH. Now, this reaction, the reduction, is coupled to this oxidation reaction, the glycolysis itself. So glycolysis is a series of oxidative steps, and NAD+ is being reduced. So this is redox or oxred. I wrote it the other way. So this is a redox reaction. Okay? Notice here that this is a reactant and this is a reactant. They are both necessary for this to take place. We need both of these for the glycolysis to take place. Both of these are reactants because both these reactions are coupled reactions. So you need both of these reactions to take place together. If glucose were to be oxidized, something has to be reduced. Okay? It's like you cannot lose money unless somebody gains it. Okay? Money doesn't just disappear. If you lose money, the store that you paid the money to is gaining it. Same thing here. Okay? So glycolysis takes place in the cytoplasm. It is a redox, and NAD+ is a requirement for this reaction. You need this. Okay? The next step is pyruvate oxidative decarboxylation. So this reaction occurs in the mitochondrial matrix, and it produces 2 NADHs, which means 2 NAD pluses are being reduced to NADHs. Actually, let me just put the 2's here so that we stay consistent. All right, so we have reduced 2 NADHs, okay, and this is an oxidation. So pyruvate to acetyl-CoA is an oxidation, NAD plus to NADH is a reduction of this molecule. Now, the third step is the Krebs cycle, which also occurs in the mitochondrial matrix. This reaction produces 2 ATPs, 6 NADH, and 2 FADH2. Okay? Now, acetyl-CoA is being oxidized to CO2 at the same time that NAD plus is being reduced to NADH, FAD is being reduced to FADH2. All of these are redox reactions. This first step, the glycolysis, occurs in the cytoplasm, and this step occurs, or these two steps occur in the mitochondrial matrix. And again, keep in mind that all of these are coupled reactions, which means for the pyruvate to be oxidized to acetyl-CoA, we need NAD plus to be available so that we can reduce it to NADH. Okay, this is a reactant as well as this. Okay, so we need NAD plus. Now the number of NAD plus in the cell is limited. They're not unlimited here. Okay, you need NAD plus. There's a limited number in the mitochondrial matrix, and there's a limited number in the cytoplasm. The NADHs and the FADH2s that get produced in the matrix, so in the second and third step in the mitochondrial matrix, these ones get oxidized in the electron transfer chain. Only. This is the only way they're going to go back to NAD+. Okay, so this happens only in the electron transfer chain. If the electron transfer chain is not working, then the NADH is going to stay NADH. The FADH2 is going to stay FADH2. And eventually, we're going to run out. Okay? Now, the NADH that's made in the glycolysis normally also gets oxidized in your electron transfer chain, but there's another thing the cell can do to oxidize it, which is fermentation. Okay? Now, why would the electron transfer chain not work sometimes? Let's take a look. Now, fermentation is a process that would regenerate NAD+ in case of anaerobic conditions. The oxygen in the electron transfer chain is the final electron acceptor. Actually, let me show you a picture here. This is from a video that I explained to you earlier. So this is the electron transfer chain, okay? And the NADHs and the FADH2s are made here in the mitochondrial matrix. They go and they get oxidized. So you see them here, they're going to the proteins on the inner mitochondrial membrane, and they're being oxidized back to NAD+ and to FAD. Okay? So they're dropping the electrons, and the electrons are going from one protein to another to the last protein here, and they're given the electrons to the oxygen. Okay? So oxygen is required. It has to be there so that it can take the electrons away. If oxygen is not there, then the electrons are going to be stuck here. And then this one is not going to be able to receive any electrons. And then the whole process is going to be backed up if you have no oxygen. And this is what happens in low oxygen conditions. So what is a low oxygen condition? Imagine your leg muscle and you're running so hard. And your leg muscle is going through glycolysis, a PDC, Krebs cycle to produce the ATP that the cell requires to keep contracting while you're running. Now suppose that your muscles are working extremely hard and your aerobic condition is not the best. Your respiratory system and your circulatory system are not going to be able to give the muscle cell, all the oxygen it needs. So now the muscle is suffering anaerobic conditions. It's not getting enough oxygen. In that case, the only thing that can take place in low oxygen conditions is glycolysis. So glycolysis is the only process that can take place during anaerobic conditions. All right? And fermentation is a very important reaction that has to go along with glycolysis for glycolysis to keep occurring. Why is that? Let's take a look. And here I explained there's two types of fermentation. Let's take a look at alcohol fermentation first to show you what I mean. So alcohol fermentation occurs only in plants and, well, it doesn't occur in humans. It occurs in organisms like plants and yeast. Now, the only process that can take place under anaerobic conditions is glycolysis. And glycolysis, at least it produces 2 ATPs. Your body cannot run out of energy. Your cells cannot run out of ATP. You need ATP. So your body just keeps running glycolysis. And as you run glycolysis, you're making 2 pyruvates and 2 ATPs, and you're also making the 2 NADHs, right? You're taking NAD plus and you're converting it to NADH. And I remember from earlier that this reaction, the glycolysis, along with the reduction of NAD plus, these two reactions are coupled. Okay, they happen together. You need glucose and available NAD plus. But eventually, if you keep running glycolysis, the NAD plus is going to run out, because in anaerobic conditions, the only process running is glycolysis and not ATC, not Krebs cycle, nothing that would regenerate the NAD plus. So in this condition, we have to run fermentation, which is these two reactions here. This is the first step of alcohol fermentation. You take the 2 pyruvates and then you decarboxylate them to make 2 acetaldehyde. And then the 2 acetaldehydes go through the second step of fermentation to make 2 ethanols. What's really important to keep in mind here, or to remember from this step, is that as the second step of the fermentation is taking place, the NADH is being oxidized to NAD+. So here, this is an oxidation reaction here. You're reoxidizing NADH to make it NAD+, and that means that this step is a reduction here, so a reduction. But we are regenerating NAD+. So the glycolysis is using NAD+ to make NADH. The fermentation is taking the NADH made and remaking it into NAD+. So this is what happens in alcohol fermentation. Now remember, your body thinks, or your muscles think, that at least, even if we're making only 2 ATPs from glucose, it's better than nothing. So it has to keep running it. Now, what happens in humans? It is almost the same thing, okay, the same purpose. Fermentation is there to regenerate NAD+, okay? But we do it differently. Animals do not do alcohol fermentation, or humans. We do lactic acid fermentation. So instead of getting alcohol, we get lactic acid that causes a lot of pain. So here we have glycolysis, glucose being converted to pyruvates. And this coupled reaction with it is fermentation. So the 2 pyruvates go through one step to be converted to lactates, or lactic acids, depends on the protonation state. In this step, in this reaction, the NADH, again the 2, are being converted to 2 NAD pluses. So glycolysis is using NAD plus, fermentation is regenerating NAD plus. Okay, so the purpose of fermentation is to regenerate NAD plus, so that the cell can keep running glycolysis, so that out of every glucose, you make at least 2 ATPs, because you cannot stop everything when there's low oxygen conditions. We have to do something to maintain our energy input. So what happens to the lactic acid in humans? You worked out really hard, you have a terrible burning sensation in your legs, and you know that lactic acid has been produced. What is going to happen to lactic acid? Does it last forever? Well, of course not, you know from experience. The lactic acid gets mobilized from the cells into the blood. The blood brings it to the liver, and it goes through something called the Krebs cycle to be converted back to glucose sometimes, or sometimes it just goes right back to the Krebs cycle. Alright, so I hope that this video helped, and if you have any more questions or any questions at all, just put them in the comment section. Bye!